go and eat. So what about the circuitry involved here? Well, here's our own old friend, the arcuate nucleus that I showed you earlier, and we see FOS activation here again. And again, this is the animals that have been exposed to the peanut butter cue that are familiar with its taste. And we actually screened the whole brain, and I have to say this was the only area that clearly popped up in our studies where we saw a big activation in response to the peanut butter cue, and we've quantified it in this graph here. So the arcuate nucleus in the hypothalamus, curiously, pops up as a very important area for food cues. We went on to try to figure out what these cells are, and we've used RNA scope, where you can do maybe 3 different in situ hybridizations, essentially, on a single section. So in this particular section, we've looked at a lot of different sections, and we have approximately per section about 80 HRP neurons, about 120 ghrelin responsive neurons, and about 100, say, FOS positive neurons. And then when we looked at co-localization, we were a wee bit surprised to discover that only 20% approximately of the cells activated by the food cue that express this FOS could be identified as these orexigenic AGRP neurons in this region. And vice versa, of the AGRP cells, not many of them were activated by the food cue. When we looked at the ghrelin receptor, this is interesting because this information comparing FOS and ghrelin tells us whether the same cells are activated by the food cue as by ghrelin, so by an external and an intrinsic orexigenic stimulus. And again, a little bit to our surprise, the numbers were not higher, but we still saw something like 30 to 40% of the cells were co-expressing cue-induced FOS and the ghrelin receptor. This is just confirming essentially some old data where we know that most of the AGRP cells in this region express the ghrelin receptor, and of the ghrelin receptor cells expressing AGRP, yeah, it's slightly over half. So if it's not AGRP, what else could it be? And this is just the statistics again for AGRP. I've just shown you about 20% are AGRP cells. We looked at another obvious candidate group in this area, the pro-opiomelanocortin cells. These are actually, conversely, an anorexigenic cell group in this area, and we found about 40% of those cells are ghrelin responsive. And finally, we did a triple RNA scope for FOS, tyrosine hydroxylase that signals the dopamine cells in this area, and the ghrelin receptor, and we found that only about 12% of the cells activated by the food cue were this tyrosine hydroxylase population, and most of these were ghrelin responsive. So around something like a third then of the cue-activated cells appear to be ghrelin responsive. So conclusions then, I think I would like to have shown you more data, but we certainly have plenty data backing this statement that ghrelin is both a hunger and an appetite hormone, and therefore intrinsic signals such as ghrelin and external sensory food cues drive food intake and food-motivated behaviours. The arcuate is targeted by converging orexigenic signals, so about a third of the cue-activated neurons are ghrelin responsive, and vice versa. The cue-activated arcuate cells are heterogeneous. They include both orexigenic cells, anorexigenic cells, and some dopaminergic cells, but we still don't know almost what a half of the cells are that are activated by the cue. And finally, external sensory food cues trigger ghrelin release, perhaps gaining its status as the aperitif hormone or perhaps an appetizer hormone. yeah, appetite and hunger may not be so different. After all, if you think of these rats in that cage going crazy, trying to get the hidden food, what are they doing there? Is this behavior because they're hungry? Or is it because they have an appetite for peanut butter? Very hard to distinguish between them. And of course, the fact that we have some convergence of signaling suggests that appetite and hunger may not be so different. And we've provided a biological basis that food cues make you hungry, releasing ghrelin and engaging so-called hunger pathways. So coming back to my original slide, I think the fact that the arcuate nucleus is targeted by food cues means that we could also place an arrow from perhaps more reward areas back to at least the arcuate nucleus in the hypothalamus. So on that note, I just thank my lab, my collaborators. For this study, especially Fiona Perez San Pedro is the driving force of this project, and Iris Stoltenberg, a PhD student in the lab. Thank you.
Thanks a lot. That was really a good appetizer for the day, and I myself have a lot of questions already, but we will leave them for the end of the session. Now use the app, send your questions, and it would be really good if you could also put in brackets who you want, an answer from, so that I can. That will be easier for me to then ask each speaker. Then we go on without further ado, and the next speaker is Aniko Korosi, Associate Professor at the University of Amsterdam, and she will talk about the entrée, the long-term effects of micro and micronutrients on cognition and stress resistance. Thank you.
Right. Thank you so much for the nice introduction, and I'm very happy to be here. It's really nice to finally give again presentations in person. It's great. This one. So we will switch a little bit gears, and I will talk to you about the importance of the early life period, and of course, the importance of nutrients during this early life period. I think I don't need to convince this audience that the first 1,000 days are key determinants of our mental health later in life. And with the first 1,000 days, we often encompass, of course, from conception up to about the first 2 years of life. And indeed, when adversities occur during this sensitive developmental period, then this has negative consequences for the vulnerability to develop psychopathologies later in life, such as cognitive deficits, depression, but also many other psychiatric disorders. So when we look into the epidemiological literature about what leads to these negative consequences, what is striking is that, of course, the adversities can take many, many different forms, often more psychological, for example, like neglect, but also physical, like maltreatment, but also in the form of malnutrition, for example. And when we look at the phenotypes that are being found later in life in these individuals that are exposed to these adversities, they are remarkably similar. In addition, it's nice to realize, or it's important to remark, that these psychopathologies are really often comorbid with metabolic disorders, and that indeed these early life adversities also lead to these metabolic disorders themselves. So there is a lot of interaction and intersection that it's definitely worthwhile to explore further. So in the lab, we really try to understand what are then the key elements on this early life environment, which can be so diverse, determining these phenotypes, which are then quite similar with each other, and which are the main neurobiological substrates, and how do these different elements interact. So some years ago, we started to think about the different interactions and the different components, and we decided to focus on the interaction between stress and nutrition, and how these early life components, the early life stress with early life nutrition together might interact with each other and lead to alterations in brain plasticity, and thereby also in the phenotypes, in the behavior, in the cognitive functioning, but also, for example, in anxiety-like behaviors. And how will these two interact? Also, of course, with in mind the idea of finding some targets that are actually possible to implement also in actual human populations. So what we use in the lab is a mouse model of early life adversity, which is of limited nesting and bedding material. It consists of exposure for the first postnatal week. So it's only 1 week of exposure to an impoverished environment. So the mother is consistently present, but she simply has like a more erratic way of taking care of her pups because she doesn't have all the facilities that the other mom has. She has less bedding material and no nesting material. And we know that this leads to altered plasticity. This has been shown by us and by many, many other labs that use actually this same model. And it leads also to cognitive deficits and changes in anxiety-like behaviors. So starting from this, we've posed the question, are nutrients then involved? Can nutrients be involved? And if so, then how? And can we use this as a possible tool or target for interventions? So why nutrients? And of course, what attracts me a lot about nutrients is that, of course, they're building blocks of all our membranes and all our cells, but they're also very important signaling molecules. And this is the aspect that I actually would like to tackle with you today. So it's important to realize that, of course, I'm going to talk about a really hardcore preclinical model, but there has been a really increasing interest in nutritional psychiatry, in how we can use nutrition to modulate actually mental health. And what we realize more and more is that while this is really attractive and it's really promising as well, and most of the studies are so far associative, and we still need a lot of information on the causality between them and the underlying mechanism. So to be able to really take all the opportunities from these kind of strategies, we need to learn a lot more about it. ECMP has also put a lot of attention to this. Indeed, we have also the ECMP Nutrition Network, which is a really specialized network focusing specifically on these questions. So the questions that I will be specifically tackling is, okay, does early life stress alter nutrient levels in the brain? Can nutritional intervention prevent these early life stress-induced effects? And then which are the mechanisms for that? So the students in the lab that actually did all of the work that I will be presenting are Dr. Kitty Yam, Kitty Reims, and Marilyn de Abing. And later on I will also mention a collaborator. And the concept is just that. We have early life stress. We have some known phenotypes that we already have characterized earlier. Can we understand what is the role of nutrients in this? And can we intervene with nutritional interventions? Then, of course, the next question, which nutrients are we going to tackle? And the ones that we decided to focus on are essential nutrients. So nutrients that we need to take in from the diet. And we focus on essential fatty acids and essential micronutrients. So let me start with the fatty acids. We know that fatty acids, I think you're all familiar with the importance of taking omega-3, eating salmon and nuts and olive oil instead of other forms of oil, for example. But actually, what is really important to realize is that the ratio of this omega-6 and omega-3 fatty acid is what is really, really important. It will be key determinant of brain development, of properties of your membrane, for how, for example, membrane fluidity is, of neurogenesis, and it has really important inflammatory modulatory roles as well. So earlier, about a few years ago, we have shown that an early diet intervention from postnatal day 2 to postnatal day, I don't manage to move the mouse for some reason, but doesn't matter. Oh, yes, now it moves with me. Now, in any case, we have this early life stress exposure, and we have the diet from postnatal day 2 to postnatal day 42. And we have shown, first of all, that early life stress actually reduces or changes fatty acid composition of the brain, which is interesting by itself. That this early dietary intervention manages to rescue these fatty acid alterations. What you see here, this is moving by itself now. Okay. And we have shown that this early life dietary intervention is able to prevent the cognitive deficits that we have induced by the early life stress. This is here shown with the object location task, but we have done that also with other tasks. And also some of the changes in brain plasticity that we have observed earlier. So the changes in adult neurogenesis, it was in the form of neuronal cell survival, but also changes in the neuroimmune cells, in microglia, in the CD6 microglia CD68, which is a phagocytic marker of microglia. So what you see in these three graphs is indeed, I don't know why this is moving by itself now. I will go back 1 second. So what you see here is that early life stress indeed leads to deficit